All right, excellent. So good afternoon, and today we are going to go over lecture 14, which is the first of the systems lectures, as I like to call them, physiological systems or biology. How do the fish work? And we're going to talk today about that. Today's topic for the syllabus is oxygen, metabolism, and energetics. And we're also going to go over respiration. And so there's a little bit of variation here amongst the different fish groups. So again, keep that in mind as far as the taxonomy portion of the course, because we'll be talking about those differences today, because not all fish operate the same. As far as the circulatory system, generally, it's all plumbed relatively the same. You know, these things have a closed circulatory system, meaning that, I get my pointer here, all of the blood, for instance, is contained within some kind of vessel or a pipe. And the fish, like other vertebrates, have a heart. In some cases, they have multiple hearts. There are also then gills for oxygen exchange. As we learned before, lungfish have lungs, and so some species of fishes actually have lungs as well as gills for oxygen exchange. But generally speaking, unoxygenated blood flows through these tissues that interface with the environment, where oxygen then is picked up, carbon dioxide then is gassed off, and the oxygenated blood then circulates to the tissues then which need that oxygen for bioenergetics. So a couple take-home things here. You'll see this in the talk. When I abbreviate O2, that means diatomic molecular oxygen, right? Oxygen exists, molecular oxygen exists as two oxygen atoms attached to one another. Arteries are vessels that lead from the heart. So an artery goes from the heart to some structure. Sometimes people think arteries always carry oxygenated blood. That's not correct, because the arteries coming right out of the heart here before they reach the gill, for instance, is unoxygenated blood. The veins are vessels that collect back to the heart. So it's really about directionality and where the heart lies in that proximate directionality. Oxygen poor blood is pumped then from the heart, typically then to a structure that picks up oxygen, whether that be a lung in, say, the lungfish, or in, say, the gars or the bowfin or other organisms which actually can use the physostomous swim bladder to function like a lung, or for most fishes, the gill. Blood is then oxygenated at these structures, and then it passes into capillary beds. And these capillary beds then provide oxygen. They distribute it really to tissues. Oftentimes when you see models like this, it's showing just generalized distribution of the blood, but it goes, you know, the muscle of the fillet. The muscle needs oxygen in order for the fish to swim. The organs, the liver, things like this. And so that oxygenated blood then is distributed to those tissues that require it. And carbon dioxide then is picked up, oxygen is offloaded, and the carbon dioxide then is transported in the blood through the heart then back to the gills or the lung, where it's released. And we're going to talk about how that occurs later today. So the fish hearts, there are two basic designs here. They have what we call the bony fishes. So again, from your notes, these are teleost fishes or osteichthyon fishes. The heart of these fishes differs from that of the elasmobranchs or the chondrichthyon fishes, the holocephalans or the whole-headed rat fishes, and also then the lampreys of family Petromyzontidae. So the bony fishes, the teleost fishes, have what we call a two-chambered heart. There's two contractile chambers. I mean, you might call this a four-chambered heart, but not in the sense of our four-chambered heart. Really, they have only two contractile chambers. They have this what we call sinus venosus. And so this is blood coming into the heart. So again, unoxygenated blood from the veins to the heart collect in this kind of muscular chamber that we call the sinus venosus. And it's very elastic and muscular. And the blood from the venous system collects in here and then passes through a one-way valve into this contractile chamber called the atrium. The atrium is contractile, so that means it can contract. It's muscular and can contract. The sinus venosus does not contract. It just is kind of rubbery and elastic. Most of this collection of blood from the sinus venosus into the atrium occurs from back pressure of the venous circulatory system. So as the fish is swimming, it generates pressure in the vascular system. And also then the forward pressure on the capillary beds from the arterial system, which is pressurized, also allows this blood to collect here and drain then into this ventricle. Now, any remaining blood in the atrium then is ejected into the ventricle with a little bit of a contraction. It's kind of what we call an atrial kick. And so the blood then travels this way through pressure, and also this atrium contracts a little bit to eject blood into the ventricle. Now, when the blood is here, this is a very thick wall. You can sort of see all this spongy myocardium. This is heart muscle. And this is a very muscular compared to the atrium and the sinus venosus. And when this thing contracts, it can generate a tremendous force. Really, it ejects blood out into the arterial system, which then remains pressurized throughout because of the stored energy in the musculature of the arterial vessels. This thing contracts very strongly, and that pressure then is maintained throughout the system by the muscular arteries. They themselves don't contract, but they sort of, if you think about it, the artery is like here, and once the ventricle contracts, it pressurizes that. Those arteries swell up, and then they slowly push blood through the system. Very active species like tunas we talked about that swim a lot. Again, these fish that have very well-developed swimming mechanisms have very muscular hearts that allow the blood to flow. Also, there's a one-way valve here in the bulbus arteriosus, which prevents backflow. And so the bulbus there is just this first arterial collection spot that, again, becomes pressurized. It sort of swells up. This valve closes and prevents blood from backing up into the heart. So the arterial system is pressurized. And this is the bony fish heart, the teleos fishes. Now, the elasmobranchs, generally speaking, the sinus venosus, the atrium, and the ventricle are all pretty much the same. The key difference here is that chondrichthyan fishes, holocephalans, and the lampreys, they have, instead of a bulbous arteriosus, they have what's called a conus arteriosus. Now, the idea here is that this sort of holds pressure and prevents blood from getting back into the heart. In this case, there's a series of valves here. In some species, as many as 72 or more valves that all then store this potential energy of the blood, this pressure of the blood as it's being ejected from the ventricle. And it passes through these valves. It's kind of like gateways. And they cannot backflow back into the heart. And so the conus arteriosus is slightly different functionally. However, its design, it uses these valves to do the same thing. Prevents blood from backing into the heart, but also then maintains pressure in the arterial blood system. So key anatomical difference, but functionally, they're the same. So that's the structures of fish hearts and where they differ. And we also talked then about the hagfish have multiple hearts throughout their body, and so refer to that lecture where we talked there about the hagfish hearts. So on the very first lecture, I gave a slide where we talked about the difference between terrestrial and aquatic life. And this is where it really comes home and is very important when we talk about respiration and ventilation. So here are some key reminders from that very first lecture. First, water has 1% oxygen by volume compared to 21% oxygen by volume that we're breathing in the air at sea level. Okay, we got a very easy job. It's easy for us to extract this oxygen from the air. Very tough to pull oxygen out of water. Second, water has a viscosity 840 times greater than that of air. It's 840 times as dense or is viscous or thick as air, and it is 60 times more dense than air, which means we can easily breathe air and pass it into our lungs and exhale it. It doesn't take much energy. Like I said, the diaphragm of an adult human being is only a centimeter or so thick, whereas fish have to have lots of muscles dedicated towards moving water across the gills. It's much more dense and viscous. More energy is then required to move water over the respiratory surfaces in the aquatic realm versus us in the air. So they have to have a lot more dedication here. In fact, 10% of the oxygen that a fish pulls out of the water, it must use just to power its breathing muscles. So when it pulls the oxygen out of the water and passes that water over its gill, it's already spent 10% of its energy just doing that. Very costly. And again, it's because of the physical properties of water. So when we think about that, that is a major consideration. The other consideration is the outright physical chemistry of water. So I'm going to use this term dissolved oxygen. So we're going to talk here about chemistry, inorganic chemistry, partial pressures of gases in solution. I know it's scary, I'm sorry, but we're going to make it painless. So all gases, you know, some gases like to be dissolved in water and other gases don't. It just so happens that oxygen's one of those gases that really doesn't want to be in water. You know, carbon dioxide likes to be in water. Oxygen, not so much. So part of the reason why it's only 1% the oxygen content compared to air. Oxygen would much rather be in air, for instance, than in water. So when we talk about water chemistry, we talk about dissolved oxygen. And this is typically reported as milligrams of diatomic oxygen per liter. In some places, you'll see this acronym, ppm. And I think I talked about this again at the beginning of class. This stands for parts per million, right? Mgs per liter is the same as parts per million. So there's a table I give you here from this reference. And so if we look here, milligrams of oxygen per liter of fresh water versus milligrams of oxygen per liter of salt water. And you'll notice something. This is water temperature. This is fresh versus salt. Now, from the very beginning, 14 mgs per liter at 0 degrees in fresh water versus only 11 mgs per liter in salt water. As the solute concentration increases, the dissolved oxygen concentration of a solution decreases. That means marine environments typically always have lower dissolved oxygen than a freshwater environment. Okay, that's a problem for fishes that live in the ocean, right? Freshwater fishes, they get a little bit easier time here because their aquatic environment is typically going to have higher oxygen content at any one given temperature. Second, as temperature increases, dissolved oxygen decreases. So right at around freezing in fresh water, we have water containing 14 milligrams per liter of oxygen. However, at 30 degrees centigrade, which is a little over 80 degrees Fahrenheit, it's half that. And look at here, same with salt water, 11 to 6. So what this means is from 0 to 30 degrees, a 30 degrees centigrade shift, we have half the dissolved oxygen. So what that means is warm water fishes have a more difficult time dealing with dissolved oxygen levels compared to cold water fishes. Cold water fishes generally have enough oxygen in the water, they can pull it out without problems. Warm water fishes, however, have this issue. And to be honest, the ones that get sort of screwed on the deal are all of the poor fish in the tropical coral reefs. There is less oxygen in warm salt water compared to cold freshwater. So the warm marine tropical fish have the hardest time with this. All right. So again, we look at the biology of fishes, their habitat determines a lot regarding their ability to deal with dissolved oxygen in the environment. So generally speaking, there's a couple ways they deal with this. The first is the methods of passing water over the gills. And so we're going to talk here about countercurrent exchange in a minute. If I were to take a fish and put it in the water and you observe it, it is going to pump water across its gills. And this is called buccal pumping. It's going to pull water in through its mouth into the buccal chamber here, and then pass that water into the opercular chamber where it's going to pass over the gills, and then it's going to come out. And we talked about how some bony fishes have this operculum. In the case of the elasmobranch chondrichthyan fishes, it's a flap or a fold, but there's going to be some excurrent siphon here. Water comes out. Now, fish can actually pump water in both directions. They can actually suck water across the operculum into the opercular chamber and then through the mouth. They can actually reverse this if they want. But if you were to observe a fish, they only do this if they're trying to cough something out. Generally speaking, they're always pulling water this direction. And that's because of a countercurrent exchange system inside of the gill. If a fish pulls water in its mouth this way and passes it over the gill, it can extract about 80% of the oxygen from that water. However, if it passes water this way, it can only extract about 10% of the water over the gills. And why is that? It's because of a countercurrent multiplier. The blood flow of the gill is in the opposite direction of the water flow, and it's plumbed that way. You're not changing the way the gill's structured. Therefore, if you change the way the water flows across that gill, you end up with a less efficient extraction mechanism. So here's a video that sort of demonstrates this, because really it's easier to see this in real time.
In this short video, we're going to look at an example of form relating to function in biology using countercurrent gas exchange in fish gills. So, we know that fish use gills to extract oxygen from the water and to release their carbon dioxide. When we look here at this picture, we can see someone lifting up the operculum of a bony fish, and we can see this gill arch with these bright red gills on there. And these gills are so red because there's lots of blood vessels running through them. And these highly folded, very thin membranes make for a very good gas exchange surface. So what we're going to need to do is have a little X-ray vision to look inside of our fish here to see the 4 bony gill arches that we see right here. And those gill arches are supporting this very highly folded thin membrane of the gills, which I'm representing by these red squiggly lines here. And so we have an opportunity to design or look at the design of how the fish gills work. Water enters into the fish's mouth, moves across the gills, and exits the back of the operculum. And as the water goes through, oxygen is extracted from the water. So I'm going to put some numbers to this just to make it a little easier to understand. I think it really helps. And I've picked the numbers kind of arbitrarily, but it'll make the point. So imagine that the water coming into the fish's mouth has 100 parts per million of oxygen. By the time it reaches the end of the gills, it's down to, let's say, 80. So we've extracted 20 parts per million of oxygen out of the water diffused into the blood. And now I'm going to let you be the bioengineer. You have the option A, to send the blood into the gills from the front, so we could come in and go through here from the front and exit the back. Or you could have the option of coming from the back, option B, sending the blood through the gills this direction and out the front. So the question is, Which would you choose? Now stop the video and think about which would be the better option. The problem with this is you know the name of this video. If you were in my class and I were teaching this, I wouldn't talk about countercurrent till the very end. So knowing the answer is one thing, but understanding it's a whole nother thing. So let's look at the why. So let's isolate this system from our fish, move the fish out of the way, and zoom in and get a little closer. Now one of the things we have to know is that gases tend to move down their concentration gradient. So if we bring our blood in this direction, choice A, and the blood's gone throughout the whole body, so by the time it reaches the gills, it's fairly oxygen poor. So I'm going to pick the number 60 parts per million. So as the blood and the water meet each other, there's a favorable gradient which allows the water or the oxygen to move and diffuse out of the water into the blood. But by the time we get to the end and we've picked up 20 from the water, the blood is at 80 and the water is at 80. So our difference between the two, we could reach an equilibrium. Now, I did pick these numbers at random, and I could have just as easily, or arbitrary, not at random, I could have just as easily made this number 50, and then by the time we gain 20, we'd be at 70, and we'd still have a favorable exchange rate from the 80 to the 70, but we certainly would have a much less steep gradient, and we reduce our rate of exchange. So let's turn the system around and bring the blood in from the back of the gills and have it run countercurrent to the water. So again, the blood's coming in, we chose 60 for the blood coming into the gills, and we have a favorable exchange gradient between the 80 and the 60. Our most oxygen poor blood is meeting our most oxygen poor water, but the water still has more oxygen than blood. And by the time we get to the end, our most oxygen-rich blood is meeting the freshest water to ensure a favorable gradient throughout this entire system. This countercurrent exchange gradient, or exchange flow, maintains the most favorable gradients for gas exchange. So we can put the system back into our fish, and we see that the countercurrent, the blood flowing from the back of the gills towards the front and the water flowing from the front towards the back, is going to give us the most favorable exchange gradient. And this is a very good example of form relating to function in a biological system. There are many others. For example, when we look at the heat exchanges in the arteries and the veins, and also water conservation in the loop of Henle. So these are things that will come up pretty frequently in biology. Alright, so if you have any questions, watch the video again, leave me questions in the comment sections below, and I hope that...
So that there is the countercurrent exchange system, and it's very similar there between different fishes, whether they're bony fishes like teleosts or whether they're chondrichthyan fishes such as sharks. Other fishes that are active swimmers, including sharks and things like tunas, that swim very quickly, energetically. You know, again, we talked about swordfishes, you know, the family Istiophoridae. These things often do what is called ram ventilation. So when they're swimming, their mouths will be open as they swim. And so instead of using a buccal pump to move water physically over the gills, they literally just open their mouths up as they swim, and water then passes into their mouth over their gills. So it's almost like a jet. So these things constantly swim, do so with their mouths open, and water then passes over the gills right through their mouth like a jet stream. So they don't have to physically pump water through it. They're using their body to move through the water like a rocket, and then just open their mouths, and as they move through the water column, their gills become oxygenated. So sometimes you'll see this behavior in very active fishes. Also, because they're so active, they also require a very high amount of oxygen. So when we look at buccal pumping in fishes, we talked about pipette feeding and how the jaws are designed to create this tremendous negative pressure in the advanced fishes like the largemouth bass and Order Perciformes. A similar mechanism is used to pass water over the gills. So when the mouth is open, water passes into the buccal cavity, and this is because the buccal cavity itself expands a little bit, and the opercula sort of expand as well. This creates a slight negative pressure, you know, nothing that's going to be a dramatic feeding kind of response, but just enough negative pressure to pull water into their buccal cavity. Kind of like you inhale air, but you're not really doing this. You sort of just gently inhale. And then the mouth closes, and the water that is then contained here in the buccal chamber then is passed into the opercular chamber by contracting this buccal chamber. This then forces the water over the gills, which are shown here, and the opercular chamber, and then that water passes out through the operculum here, or the excurrent siphon in the case of some of the more derived bony fishes like the puffers. So expansion and contraction of the oral and the buccal and opercular cavities ensures that there's a negative pressure here and then a negative pressure out here, and then that passes water then from the external environment into the buccal chamber, into the opercular chamber, and then back out into the environment. And this process repeats over and over again. Left, right, left, right, left, right. So here's a video that sort of demonstrates that in a typical fish. And so it's sort of, it shows this sort of in real time. You can sort of see it.
Unlike land animals, almost all fish breathe or get their oxygen from water. Instead of using lungs, fish breathe with organs called gills. Most fish have 4 pairs of gills enclosed in a gill chamber on each side of the head. On the outside of the body, a flap of bone called a gill cover protects the gills of bony fish. To get oxygen, fish gulp water through the mouth and pump it over the gills. In many fish, the breathing process begins when the gill covers close and the mouth opens. The walls of the mouth then move inward. The mouth partially closes and the gill covers open. This action forces the water from the mouth into the gill chambers. The inside of each gill consists of fleshy, thread-like filaments. Water from the mouth passes over the filaments. The filaments are closely spaced along a gill arch in 2 rows. Each filament has many tiny extensions called lamellae. Blood flowing through the lamella takes oxygen from the water and releases carbon dioxide into the water. After the fish receives its oxygen, the water passes out through the gill openings and the breathing process begins again.
So that that there then shows that also, again, you can see at the end there the countercurrent exchange. The blood flow is the opposite direction of the water flow. So countercurrent exchange. But again, the buccal pump and the opercular, the buccal cavity and the opercular cavity both then exchange turns being the source of the negative pressure. And that sort of then allows the water to pass through this system. Okay, so that's buccal pumping. And again, fish that do ram ventilation, they can do this if they're at standstill, but the other way to do is just to open your mouth and swim really fast through the water and just let the water naturally pass through this system, right? That's the ram ventilation. So elasmobranchs are similar in their ventilation. They also have a buccal pump, and they also can perform ram ventilation, especially in the fast swimming sharks. Major difference is, according to the first video that we showed, there's 4 gill arches in a bony fish. But remember from the very beginning of lectures, the elasmobranchs, a typical elasmobranch, it has 5 gill arches. So 1, 2, 3, 4, 5. Some chondrichthyan fishes have 6 or 7. But generally speaking, there's 5. Bony fishes have 4. Also remember from Romer's hypothesis and the invention of jaws, there can be a spiracle located here between what would have been the 3rd and 4th gill arches. And bony fishes, of course, then have a bony operculum. Chondrichthyan fishes, of course, have no bones. They're cartilaginous fishes. They have these kind of flaps here. They have gill slits, and that hence the name elasmobranch, the elastic gilled fishes. So some major anatomical differences there between chondrichthyes and teleost bony fishes. And again, some bony fishes do have a spiracle, but primarily, you know, it's a key feature for some taxonomic groups. But chondrichthyes have this, and the spiracle is a 2-way valve that's also involved in buccal pumping. So, for instance, a chondrichthyan fish can pull water into its buccal cavity and then pass it through this branchial cavity, because again, they don't have an operculum. It's a branchial cavity where the gills are, and then the water then passes out here. But they can also pass water in through this spiracle in addition to their mouth, and then pass water over the gills. And so they can actually use this as an in-current or X-current, because it's really both ways. They can control this valve. And that's because, look at the lifestyle of this creature here. If this thing was actually pulling water in through its mouth all the time, it would literally be sucking sand. And so a lot of times what it does is it pulls water in through the spiracle and then exhales through the gills, which are on the ventral side. So it's a structure that is sort of adapted. So there's a little bit of anatomical differences here based upon the life histories of these different fishes. And so here's a video there of a stingray. You can see it using this spiracle. You can see the spiracle opening and closing as it ventilates. You see, these things stir up quite a bit of sand when they're down there, and so that's why they're sort of taking water from the other side. So that there is the spiracles. So major differences there. No operculum, the elastic gills, and then also the presence of a spiracle. And again, some other fishes besides chondrichthyes have a spiracle, but that's why I always said it's primarily kind of a fancy taxonomic character. It's a unique thing. And so those are the chondrichthyan fishes. When we look at the hagfish, again, family Myxinidae, and then the lampreys family, Petromyzontidae. Hagfishes have one or more external gill openings up here. And remember, these other pores down the side of their body are mucus glands they use to produce this slime. Inside of their head, they have this velum, and then they have this nostril, this prominent nostril on their head. Water does not come in through the mouth for breathing. Only thing comes in the mouth is food straight to the gut. They actually inhale water through this nostril. It comes down here, and it's really a pumping action by this velum, which draws water in, and then it comes into these gill sacs that are down the side of the fish, and then each one of these gill sacs has an X-current opening that water then passes out of the side of the hagfish. So these X-current ducts, these are called branchial ducts. And again, I talk about this on the lecture when we talk about hagfish. Lampreys have multiple external gill openings as well on each side. Remember, like a sea lamprey is going to use this oral disc to physically attach to something. And if you're attached to the side of a fish, parasitizing it and eating, if you had to use that same mouth to breathe, you'd suffocate. So it's not a very good adaptation to use your mouth to breathe. So the mouth actually doesn't even connect really to this pharynx of the lamprey. The lamprey have these openings on the side, and each one of these openings then is associated with a gill pouch. And these pouches are muscular, and they literally will pump water in and then pump water out. Pump water in, pump water out. So these things actually can just breathe. Each one of these muscular pouches can pump water in independently. And so that way the lamprey is able to breathe even while it's attached to a host, or like the videos I showed, they sort of sometimes adhere to rocks and things like that to avoid being swept downstream. So again, quite different methods of gas exchange here. Myxinidae, Petromyzontidae are the two families. We look at the gill structure of the bony fishes, the teleosts. As I mentioned before, there are 4 gill arches, 1, 2, 3, 4. These are then protected and lie underneath this bony operculum, and they're numbered superficial to deep. Superficial means right at the surface, and deep means away from the surface. So if you open up that operculum, the very first one you see is gill arch 1. If you flip this one to the side, the next one down is 2. and 3, and then 4, and so on. There are 4 arches on each side of the head. The gill arch supports what are called gill rakers, which are these hard kind of like these spiky, bony looking things. And then the gill filaments. And I talked in the very beginning of this class about how the original gills that originally were developed in the earliest animals, these filter feeding sessile animals had these external gills that fed on stuff. And I said that later on, the gills of a fish are involved in both respiration and feeding, just like those ancient animals were, and that's what this is. The filament end of the gill is involved in feeding, or the filament end of the gill is involved in gas exchange, and the raker end of the gill is involved in feeding. So when we talk about planktivores, they literally will filter plankton with these gill rakers. So feeding, gas exchange. Same function, just inside of the head. The gas exchange occurs again countercurrent. Here are these lamellae, the video we're talking about, that sort of lie on these filaments. So here's our gill arch, which is kind of bony, and we've got blood vessels inside of it. On top of these arches are these filaments. There's 2 filaments, a left one and a right one. So each gill arch has 2 filaments on it. And then on these filaments then are these little leaf-like folds called lamellae. And lamellae is exactly where this gas exchange occurs. So these little like sort of folds on the top. And again, if water is flowing this way, blood on the lamellae are flowing the opposite direction, countercurrent. So this is just another figure showing this in addition to the 2 videos you've seen. Here's another figure then showing that countercurrent exchange. So oxygen poor blood enters the gills again through the mouth into the buccal cavity, and then from the buccal cavity to the opercular cavity. Here is our gill arch, 1, 2, 3, 4. Here are our gill filaments on the other side of those gill arches, and here are these folds, these lamellae that lie on the gill filaments. If our water is flowing this way, our blood is flowing the opposite way. So blood is flowing this way down the lamellae, water is flowing the opposite direction. This maximizes our countercurrent exchange or countercurrent multiplier. This maintains a optimal gas diffusion constant across the entire surface of the gill. So that fish is able to maximally extract oxygen from the blood. Okay, so again, just a summary here of the 2 videos. And it's a figure you can look at. When we look at fish blood, there's a lot of similarities here between fish and other vertebrates. There are some key differences as well. So I've got this slide here broken up into 2 parts, what we call formed elements and plasma, blood plasma, which really is simply the blood minus the formed elements. So we'll talk about the formed elements first. This is really where some of the key differences lie with fish. The first is fish, the first formed element, and formed element means like, this is an example blood smear of fish. Formed elements are a fancy way of saying cells or cell-like materials in the case of thrombocytes. They're enclosed, they're formed. They're not just chemicals floating in solution. These things actually have some kind of structure to them. They're formed elements. So one major difference, red blood cells in fish literally are cells. You know, people call RBCs red blood cells in a human, for instance. That's really not correct. A cell, by definition, has to have a nucleus. Well, your red blood corpuscles, as they're called, a corpuscle is a cell without a nucleus. So it's a formed element without a nucleus. So you have red blood corpuscles, just so happens it's the same abbreviation, RBC. In the case of fish, they are literally red blood cells, erythrocytes. They're a cell because they have a nucleus. These cells contain hemoglobin just like your corpuscles do, which gives them the red color. And this hemoglobin then binds to oxygen and carries it. You also have white blood cells. Leuco is white, cyte is cell, erythro is red, cyte is cell. So the leukocytes function in immune stuff. So for instance, here are some example leukocytes. We've got neutrophils and macrophages and eosinophils here, basophils, lymphocytes, monocytes, all sorts of stuff. You know, these things are involved in creating immune factors like antibodies. Also, they produce inflammatory compounds that are involved when like a fish is damaged and you need an inflammation response to help things like blood clotting, which is where our thrombocytes come in. Thrombocytes are these sort of blood clotting factors. They're similar to your platelets. And so they're kind of like these little like raggedy pieces of cells. They're like fragments of cells. And these things contain clotting factors so that when this fish is damaged, for instance, and its blood is exposed to the environment, they allow a clot to form so the fish doesn't bleed to death. So they're clotting factors, just like the platelets in your blood. So very similar. So we just call them thrombocytes. We do not call them platelets because, again, these are actually cells. When we look at plasma, there are some key differences as well. One difference. The first is this here. This is not a difference. This is the same. You, just like a fish, most of the carbon dioxide in your blood is actually transported in the form of carbonic acid. And we'll talk about that in 2 slides. Carbonic acid. It is, carbon dioxide does not bind really to hemoglobin. Oxygen does that. Carbon dioxide dissolves into solution. I opened the talk by saying carbon dioxide likes to be in solution. It can dissolve into water. There's no problem. And so there's no point in having it hitchhike a ride. The reason why we have hemoglobin is because oxygen really doesn't want to be in water and we want to transport it. And so we create something that does that because the water just doesn't do the job. Water, ions, nutrients, waste products. Waste product is the big difference here. Your body, when you break down proteins, your nitrogen waste is excreted in the form of urea. Fish excrete ammonia. So it's a different form. Urea is really like 3 ammonia molecules stuck together. Fish don't bother doing that because it's an extra biochemical step. They just say, let's just make the ammonia and get rid of it. So that's a key difference there. Immune components, again, antibodies, clotting factors, things like that, very similar between fish and mammals. And chemical mediators, hormones and other things are very similar as well. These fish have hormones just like you and I. And also they have a complement system which then goes with the thrombocytes to help the blood to clot. So other than that, really ammonia and the cell here and the thrombocytes are really the key differences between fish and mammals, for instance. So here's a video of bleeding fish. You know, a lot of times we do collect blood from fish. It's a very easy tissue to sample. And usually we do it right here out of this is the caudal peduncle. The caudal vein is right here along the backbone. And so you can bleed a fish really easy out of the caudal peduncle. So here's a video showing that.
Okay, with your fish anesthetized, we can take a blood sample. Blood samples are really good, so you can actually test for organ function, whether there's organ damage, and also the immune system, how they're going, whether they've got an infection going on. So basically you go between the tail and the anal fin, midline, and try and measure and see what sort of angle you need to get to where the spinal column is. So you think that you probably go at this angle and about this deep. Make sure your needle is actually going to be long enough to reach. So basically, stick the needle in midline until you hit the spinal column. For the ease of it, sometimes if you hold the fish completely vertical and upright, you can actually go more centrally, and that way you can actually get the blood sample a lot more easily. rather than having the fish lying on its side. Here you are. And then when you finish, put some digital pressure on the injection point, pull out your needle, and your fish should be right to go back. And then I'm just going to put this into some lithium heparin. This is great for clinical chemistry. So you notice I took the needle off the hub of the syringe first. That way you don't actually lyse the cells when you put it into the anticoagulant. And gently rock this back and forth, and try and put it into the anticoagulant as soon as possible, because fish blood does clot quite readily. And you can store that in the fridge to be collected for lab testing.
So I have bled many a fish before, and that is very routine. So it's actually it's very easy to bleed a fish, actually, right out of that caudal vein. So again, very similar to other vertebrates. We look at what we call the hematocrit. This is the concentration of those red blood cells in the fish blood, and it's usually expressed as a % of the blood. Hematocrit is greater in highly active fish species. So again, here we look at very fast swimming fish, scomber. Remember, family Scombridae, the mackerels. Look at this, hematocrit, 52. Half of the blood volume is red blood cells. That tells you how much oxygen these things need. Versus Lophius, family Lophiidae. Remember, these are the goose fishes or the monkfish. They sort of sit on the bottom, kind of like an angler fish, and they wait. This is where the fisherman got his hands stuck in the sort of just, it's like a feeding bag that sits on the bottom and waits for stuff to swim by. Look, hematocrit's only 17%. They're very sedentary, not very active. So active fish have a higher concentration of red blood cells. Hematocrit also can be used to evaluate health of fish. You know, for example, low or high values may indicate disease or stress. So they publish these factors for a lot of different species, like hemoglobin concentration, hematocrit, things like that, because these are baseline values. You know that if you've got a fish, for instance, that should have a hematocrit of 20, if the hematocrit of a fish that you measure is either greater or less than that number, maybe that fish is sick, right? It's diseased, or maybe it's stressed. If this fish is in a low dissolved oxygen environment, like due to pollution, the hematocrit might increase. And so measuring the hematocrit of different fish within the same species in different environments can be used to indicate, you know, habitat suitability, for instance, or levels of pollution, or things like anoxia, you know, which anoxia oftentimes accompanies things like eutrophication, which results from pollution. Also, I'd like to just point out, this is an interesting thing in the physiology. Thunus, right? This is the warm-blooded tuna. It's active, but unlike the mackerel, the hematocrit's quite a bit different, and this has to do with warm-blooded versus poikilotherm, and that's because oxygen has a tendency to diffuse a little bit differently with the metabolically warm animal. And so there are differences there as well. And so even though the thunus is active, its hematocrit's a little bit lower simply because of its warm-bloodedness. So interesting features there. When we talk about hematopoiesis, heme is blood, poiesis is to produce, the production of blood. Now, the earliest fishes we talked had bones. In the case of chondrichthyan fishes, these bones were lost. They're cartilages. They're cartilaginous chondrichthyes. They have no bone. In the case of other bony fishes, our trend was lose the heavy armor, reduce our skeleton, create a bunch of muscle to put on it so we can swim very fast and we're agile. Well, what that means is they've got no bone marrow, right? If you ask an intro to vertebrate biology question, where is the blood produced in a mammal? Well, the bones, the bone marrow, they produce it. Well, guess what? Fish don't have this. So if you answered that on an exam, you're wrong. As a matter of fact, the primary sites of blood formation is the spleen and the kidney, the head kidney of the fish. The head kidney, simply enough, because it's the part of the kidney facing the head, the anterior portion of the kidney. But really, it's the spleen right here, this little cherry-colored organ, and then the head kidney, the part of the kidney facing the head. The spleen also stores large amounts of red blood cells. And so if a fish actually gets injured or stressed or swims from high dissolved oxygen water into low dissolved oxygen, the spleen will literally contract and release a bunch of red blood cells like that. The fish can actually change its hematocrit in a matter of seconds. So it can go from a hematocrit of 30 or a hematocrit of 20 to 30 just by contracting the spleen. So they're kind of cool. We cannot do that. The spleen and kidney also are the sites then of immune cell production, the leukocytes as well as the erythrocytes, the red blood cells. So again, if you answer bone marrow, you're wrong. It's a major difference here between fish and mammals. They produce it in these organs here. They don't really have bone marrow. When we look at oxygen transport in the blood, the primary respiratory pigment we're talking about here is hemoglobin. Hemoglobin, I abbreviate here HB. And this is the respiratory pigment. It is what gives red blood cells their red color. It contains iron. And when iron binds to oxygen, it turns this bright red color. And hemoglobin has a very high affinity for oxygen. We talked about how oxygen's at a very low concentration in the water. We talked about we actually have to use a countercurrent multiplier in the gill just to get enough oxygen out of the water into our blood to survive. And the hemoglobin's key because it literally grabs a hold of this oxygen very tightly, very high affinity. It then takes that, it binds to that oxygen until it takes it to a tissue where then it needs to be offloaded. Now, a typical hemoglobin is made up of what we call 4 subunits. And inside of this, I've got this little red circle is the iron. Each heme has 1 iron molecule. And fish have 4 hemoglobin subunits. Now, the typical assembly is what we call 2 alphas and 2 betas. And you're like, what's an alpha? What's a beta? Don't worry about it. It just means that alpha is different than beta. And so I talked on the previous slide about how if a fish goes into from a high dissolved oxygen environment to a low dissolved oxygen environment, like it's literally swimming along and all of a sudden it notices, oh man, there's not enough oxygen around. The spleen can contract and release a bunch of red blood cells that help deal with that. And that's an immediate thing. Let's say though that you're a fish and you're living in very warm water and it's getting warmer in the summer, the dissolved oxygen's going down, and you're adjusting to that. Well, you can actually rearrange these subunits such that the affinity of hemoglobin for the oxygen changes, where you now have a higher affinity hemoglobin for oxygen. And you rearrange it, for instance, instead of doing 2 alphas and 2 betas, you might do 4 alphas. And you're like, all right, 4 alphas are more have higher affinity for oxygen. We can bind it because it's going down in concentration in our environment. But let's say then winter comes along and it's starting to cool down and the oxygen in the water is increasing. Well, we can rearrange that back. We can start producing hemoglobin then that has a different configuration of subunits that might not necessarily need that high affinity for oxygen. So fish can adapt to this. We can't. You know, if you're an athlete, you hear these guys that are like, cheating at the Olympics and stuff. They're doing what's called blood doping. All blood doping is mimicking this. I mean, fish have been doing it for millions of years. They did literally take a bunch of red blood cells and inject it in their body and increase their hematocrit. That's what blood doping is. Athletes cannot do this. Even if you're training at high altitude, all you're doing is increasing your hematocrit. You're not changing the structure of your hemoglobin. Fish can do this, mammals cannot. So the hemoglobin composition in fishes can change based on environmental dissolved oxygen. Again, when you're looking at environmental assessments, this is another indicator. You can look at actually the abundance of different hemoglobin proteins in the blood of a fish and its affinity. Pretty cool. So here, most carbon dioxide, just like you and me, most carbon dioxide is dissolved in water. Carbon dioxide likes to be in water. I don't know if you guys did this as intro to biology. If you take a cup of water and a straw, and you put the straw in the water and you blow into it, you blow bubbles in the water. If you sit there and do that for 10 minutes, you're going to decrease the pH of that water. Why? Because the carbon dioxide you're inhaling will actually dissolve into the water to create what is called carbonic acid. And carbonic acid then decreases the pH. It's an acid. And so this happens very rapidly and readily. So carbon dioxide, unlike oxygen, has a very good partial pressure in water. It wants to be in water. Oxygen, we sort of have to force it by creating this pigment called hemoglobin. So carbonic acid then will dissociate into a proton, which is what makes it an acid, right? This is literally the hydrogen ion is what you measure in the pH. And then this bicarbonate, which is the conjugate base. So carbonic acid dissociates into a proton and bicarbonate, the conjugate base. And this is what forms the buffering system for your blood, for fish blood, actually for any vertebrate blood. This is the system right here. And so carbon dioxide released in your tissues just easily does this. You don't need to bind it to something. It just, boop, chemistry. It happens. So it also is very important for blood pH, and the blood pH is very important here. So let's say we have a low dissolved oxygen concentration in our environment. Okay, this figure up here, we've got our gill here, and we got our tissues. We're going to call this muscle, fillet, whatever. The gill is here. Out here is our water, our environment we're swimming around in. And I just said, very low dissolved oxygen environment, 1% oxygen compared to 21% where we are. We have to wrestle to get this oxygen out. We use countercurrent multiplier and we use hemoglobin, very high affinity to pick up oxygen. It's going to strip all the oxygen out of that water as it passes over our gill, or in the case of a lungfish, actually, hey, it has to deal with 21% oxygen in the atmosphere. It's actually doing pretty good. But in the gill, it's still pretty low. We bind it to this hemoglobin. That's great. We've accomplished part one. Get the oxygen out of the environment and into our blood. All right, we're moving our oxygenated blood to the tissues, but we got a problem. What is that problem? Well, I just told you, hemoglobin has a very high affinity for oxygen. Do you think it's just going to let go of the oxygen? No, no. As a matter of fact, hemoglobin never wants to let go of it. It fought to get it. It's not wanting to get rid of it. So what happens here is we have to try to convince hemoglobin to drop off the oxygen at the tissues where it needs to go. And the way we do this is we use our old friend, carbonic acid, because we're going to take advantage of some chemistry here. And we're going to call biochemistry. One way we can tell hemoglobin to get rid of oxygen is by changing the pH. So oxygen is bound to the hemoglobin, and we've got tissues here that are producing carbon dioxide through cellular respiration. This is part of the Krebs cycle, tricarboxylic acid cycle. Carbon dioxide is a byproduct of breaking down carbohydrates or anything with carbon. It's the waste product. for energy production. This carbon dioxide, as I said, easily dissolves into solution to create carbonic acid. In fact, 95% of carbon dioxide transported in your blood is in the form of carbonic acid. It's in the blood plasma, it's in the cellular tissues, it's in the cytoplasm, it's in all your body fluids, carbonic acid. That means then the pH at the tissue is lower than the pH here. So if we've got oxygen bound to hemoglobin that's going into a low pH environment, we're going to take advantage of two effects: the Bohr effect and the root effect. Now, the Bohr effect is in all vertebrates. You, me, all fish, every vertebrate has what's called the Bohr effect. If it's got a circulatory system, it's got a Bohr effect, because otherwise you couldn't offload oxygen. And then fish have a special root effect. Only fish have this. No mammals do. And what this says is this: at a low pH, the affinity of hemoglobin for oxygen decreases. Hemoglobin is going to want to offload oxygen at a lower pH. What the root effect says is that not only are you going to have a low pH, you're going to have a lower affinity at a lower pH for oxygen, you physically can't bind as much oxygen. It actually causes a conformation shift in the hemoglobin protein such that it cannot bind oxygen with the same affinity, or what we call maximum binding capacity. You are forcing hemoglobin to dump the oxygen at the tissue where you need it. Now, some of the carbon dioxide will then bind to hemoglobin because, I mean, carbon dioxide does contain two oxygen molecules. And so in some cases, there's a little bit of association there, but by and large, 95% of it is just dissolved as carbonic acid, which then shifts, it diffuses into its proton and conjugate base bicarbonate. That then transfers, goes through the bloodstream to the gill, and then carbon dioxide will diffuse down its concentration gradient from the inside of the fish to the external environment. Bicarbonate will go out, the proton will go out, carbon dioxide will go out. It just goes out into the water around the fish because the environment is always going to have lower levels than the inside of the fish. That is how the gas exchange occurs. Okay, plain and simple. Very important. Our friend, carbonic acid. We'd all be dead if we did not have carbonic acid. It's the only way we can actually get oxygen where it needs to go, and the only way we can get it to offload where it needs to be. And it's about chemistry. So some other interesting things about fish. The pseudobranch, I talked about this in lab. If you open up the operculum like this, here again is a bony fish. 1, 2, 3, 4 gill arches, right? Bony fish. Here's the head of the fish. Here's our pectoral fin. And sometimes there's this little kind of flap here, this little, what we call pseudobranch. And it kind of looks like a miniature gill arch. Present in many bony fishes, small rudimentary gill arch. It's attached to the inside of the operculum. What does it do? We don't really know. Okay. We're going to talk about osmoregulation in one of our lectures. And so some people say that it might have an osmoregulatory function. So it allows us to regulate our salt base and pH balance, or at least the fishes. Others say it might have a sensory role, for instance, oxygen sensing or some other kind of environmental parameters, because there's a lot of nerves that go here. It's innervated by the nervous system. And some say it might provide oxygenated blood. There are gill filaments here. As small as this thing is, there is a little bit of gas exchange going on, and maybe that oxygenated blood goes directly to the eyes, and in some cases, the retina. And so some very visual species might specifically oxygenate blood at the site of the pseudobranch, and that blood goes straight to the eye. It doesn't go to other tissues, so it's only the most highly oxygenated blood. And that's because the retina requires a lot of energy. I mean, when you look at vision as a sense, it is a very expensive sense to run bioenergetically. This thing, I mean, think about it. Your eyes are constantly taking pictures of everything around you and then we're sending that to the brain. Very expensive sense to run. So the pseudobrain. So this is the last slide before I go to the story. And I put this in there because really it's part of the chapter reading. I think it sort of doesn't belong here. If I was writing this book, I'd have put this maybe with the digestion stuff. I don't know. But we might as well cover it. And this is bioenergetics. So you are what you eat, so to speak. There's an equation up here. C, consumption, what you put into the organism, equals E, which is the waste, excreta, plus R, respiration or biological cost. I put the word entropy here because this is energy you cannot use, but is released. You know, in physics, we call this heat. And heat, once heat's created, it just goes off into the universe and it's gone. And then plus P, which is production or net production. This is stuff that is useful. So when you eat something, there's always some part that you cannot digest. It might contain energy, but you just cannot use that energy. You excrete this. This is feces. This is undigested stuff. This is useless waste. Now, it's not useless because some other organism might use it. But for you, it's useless. So it's called excreta, the feces. Now, once we pass the rest of that stuff through our gut, we can pull stuff out of it. We can do what is called, we can digest it, which means to break it down, and we can assimilate it. Now, assimilate means it's literally taken in and becomes part of you. But that's not free either, right? If you've got stuff hanging around in your gut and you want to pump it inside of your body into the bloodstream to use it, pumping that stuff isn't free. It takes energy. And this is the energy required to take it in and also the energy required to get rid of it. So let's say, for instance, we ingest protein. Well, we use the protein. We burn it for energy. And like I said before, the carbon dioxide is produced as a waste product of breaking down carbohydrates or other carbon-containing things. It's easy to get rid of that. I blow it out. Nitrogen, you can't do that. Nitrogen, you have to create something. In fishes, they create ammonia and they excrete it. I've got urine here that's wrong. They actually produce ammonia and excrete it over the gills. This figure is correct for a terrestrial animal like you. You produce urine, and in your urine is urea. And urea literally is just three ammonia things attached to each other. So there's cost there. It's not free. Let's say then we paid that price. Now we're going to go down to our assimilated energy. All right. It's not free. We got to move stuff around. We call this specific dynamic action. Again, creates entropy. If you've got a nutrient in your liver and you want to send that to your muscle, it's not free. You can put it in the blood, but guess what? Heart's got a pump. The pump's got to move the blood around, and then the blood might get to where it needs to go. That's not free. All right. So there's a cost to transport that from tissue compartment to compartment inside of your body. It's not free. So to go from a storage tissue like glycogen or glucose from the liver or fat from your adipose, it costs stuff, energy to move that around. Then we have what's called standard metabolism. This is like cellular membrane potentials, you know, their nervous system. It costs energy to maintain this. It's not free. In order to live, we have to maintain organization in our internal environment outside of our external environment. Osmoregulation, I mentioned. We have to maintain our salt balance. This isn't free. There's a cost. And then active metabolism. Lord forbid you've got, you know, we've already gone down this list. Lord forbid another fish is chasing you to eat you and you've got to swim away. You have to evade a predator. Or you're hungry and you have to get more of this consumption, and you got to pursue something. You got to go out and get it. There's a cost to active metabolism. You're swimming around your muscles, you're using your muscles. That's not free. It costs energy. And I said before, another standard metabolism, just moving the water over your gills. 10% of the oxygen you get, you use for that. Very expensive. And what are we left with? This measly net production. If we got injured, we got to repair it. If we got sick, we got to fight off a disease. Oh, now we might have a little bit left to actually grow. We can get a little bit bigger or produce gametes to reproduce. All right. And generally speaking, depending on your environment, how happy you are, how much you're able to eat, how much stuff is chasing you around, or how sick you might get, you have between 0 and 40% of your energy then dedicate towards growth and reproduction. So it's usually, it's the equivalent of the spare change in your couch. You're trying to buy dinner and all you got is a few nickels and dimes out of your couch cushions. And so this here is bioenergetics. So with that, I have stored. And again, I like to always show these. This is a weird phenomenon in fish. You learned it here, AEC 441, the unique blood of the ice fish. The Antarctic ice fishes, family Canichthiidae, occupy the coldest, most thermally stable marine environments on Earth and provide an excellent example of the unique traits of fishes that can arise in chronically cold environments. Now, I told you at the beginning, marine water, generally speaking, has lower dissolved oxygen than fresh water. However, this is the Antarctic, the coldest water on the surface of the Earth. Actually, the water is so cold, if the salt wasn't in it, it would be a block of ice. It's below the freezing point of fresh water. Because oxygen solubility in water is inversely proportional to water temperature, the cold Antarctic seas are an exceptionally oxygen-rich aquatic environment. So they, again, cold water, higher dissolved oxygen. So in the extreme cold, there's a problem. Fluids like blood become viscous and thick. Formed elements like red blood cells actually make the blood so thick it can't be circulated. It's like oil or sludge. It's not like liquid runny blood. It's like thick sludge. Now, ice fishes are the only known vertebrates to completely lack red blood cells. They also don't even have hemoglobin. As adults, the blood appears clear or white. Here's a picture. This is red blood from a regular red-blooded fish, or like blood out of your veins. This is ice fish blood. It's clear or white. No hemoglobin at all. And if you actually centrifuge, if you try to find cells in here, there are no oxygen carrying cells. Oxygen is solely dissolved in solution in the blood of an ice fish. So it only has an oxygen carrying capacity of about 10% or less of that of red-blooded fish. Again, because the partial pressure of oxygen is such that oxygen doesn't want to be in water. But we're in a very cold environment, which favors that ever so slightly. So ice fish possess very large hearts. Look at the size of the head. We dissect a fish. The heart is usually right here, lies up right next to all those gill arches. Very large heart. And they also have a blood volume up to 4 times greater than those of red-blooded fishes. And these features collectively permit a large volume of blood to circulate through the body of an ice fish. And despite this low oxygen carrying capacity, they provide adequate oxygenation to the tissues. So, unique feature here, always surprising with fishes. You can say, you can't say they don't have blood, because they do have blood vessels and they've got this fluid in it, but they don't have red blood cells. So there you go. You learned it here, the blood of ice fish. So with that, I will end the recording and I will entertain questions. Thank you.